Good morning, everybody. Thank you for the organizing committee for inviting me for this exciting opportunity. And when I received the invitation, to be honest, I was reluctant about the topic of the generic immunosuppression and biosimilar. And I thought that it might not be relevant in the 2021 to discuss this issue. Simply because when I tried to search the PubMed and the internet sources for any recent publications about this topic, I found a scarce amount of publications to cover this area. Probably partly because it has been already addressed and there is nothing much to add, or it has been stabilized already and no need to discuss it anymore. But I realized that still there is some anxiety, particularly amongst the transplant colleagues, to initiate the generic immunosuppressive medications as a de novo, or to change those currently maintained on the branded generic, the branded immunosuppressive medications. So I thought that, okay, let us discuss it. It might need further discussion, and I would be more than happy to have, the discussions and to see whether in Egypt we are up to the scratch in terms of having these generics prescribed in the as a part of the immunosuppression or not. So the learning objectives of this talk will be: what is the definition of the innovator, generic and biosimilar, the generic approval requirements, what is the evidence behind their widespread use nowadays, what recommendations when we use them to adhere during the conversion process, if we are going to use it as a conversion or switch, and then a brief account on the biosimilar. We have to start with definitions, first of all. What is meant by innovator or originator, which is the normal drug. When you need the full registration dossier, you need usually the license, the requirements for the efficacy and safety. You need the randomized control trial, phase 2 and the phase 3 to prove this efficacy and safety. This is not the case in the generic medicine. Generic medicine has got exactly the same active ingredient, but differs maybe in the excipients, maybe differ in the salt. Say, for example, when we use the sodium bicarb, or the salt could be chloride. You remember the sevelamer, when we have got the sevelamer bicarbonate or sevelamer chloride. So the salt could differ, but at the end of the day, the route of administration is the same like the branded. The intended use, it is the same. It should be pharmaceutically equivalent and also bioequivalent. And we will come to discuss what is the requirement to approve a generic medicine. The biosimilar, it is a biological material, a biotherapeutic product, which is, as it states, similar, not exactly the same, not like the generic and the innovator. The generic and the innovator, the active ingredient is exactly the same. But for the biosimilar, they are not exactly the same. They are only similar. That's why the approval process for the biosimilar still requires the clinical trial for proving the efficacy and safety process. And that's why the biosimilar, sometimes they are still expensive to the, compared to the original biologic material. Just like the generic, when it is very cheap because you don't need all these requirements for the proving the efficacy and safety. Why we use the generic in the first place? Simply because the cost saving. And some people, in particular in the immunosuppression, they claim that when you have got cost saving and the patient pays out of his pocket, you prove and improve the adherence and probably the graft survival. We can see that 1/3 of the world's population is unable to access essential medicines, mainly due to the cost. The United States has saved around $313 billion in 2019 from the generic and the biosimilar drugs. And be careful that this is all generics, not immunosuppressive only. The Affordable Care Act in 2010 has expedited the process of generic approval by the FDA, and it allowed the prescription of the generic instead of the branded when it is available. You can see that in the United States markets, the generics constitute around 90% of the prescriptions, which only costs around 20% of the total cost or expenditure of the medicines. In Canada, they save around 27 billion, with 73% of the prescriptions are generics. And these 73% prescriptions, they only cost 20% and the 80% for the branded ones. In the UK, 81% of the prescription from the primary care are all generics. And again, this 81% was around 90% cost saving. You can imagine, this is for all disease entities, but I didn't speak about the immunosuppressive in particular with the privilege of this class of medications. How much is the savings that we do, I mean the United States in particular, to the immunosuppressive medication for the generics? You can see that if it is, for the government saving, it's for the tacrolimus is around 50% per patient per year, and for the mycophenolate is around 55 to 70%. For the patient, when he pays from his out of pocket after 3 years from the transplant, it's around 60% for tacrolimus saving per year, and it's around 70% for the mycophenolate. So it is a huge cost saving. But does this worth to use the generics and jeopardize the graft function, if we can put it in this way? We will come to this point soon. This is a very interesting comment from one of the kidney transplant patients in the same article in the same issue of Clinical JS in 2019 about her feelings of the generics and how much she has saved using the generics across the 20 years of the lifespan of her kidney transplant. And she raised some few questions and some few concerns, if we can put it, and I would encourage everyone to read this, this view from one of the patients who is healthcare aligned. I believe that she is a dental technician, something like this. Very interesting. What are the FDA generic approval requirements? If we come to this point, we can see that when we approve the generic compared to the branded, they require a crossover study for 24 up to 40 healthy participants, studying the bioavailability of the drug with the Cmax and the area under the curve for the concentration across the time to be between 80 and 125% of the comparable to the branded one, with the 90% confidence. So, this is a very wide range, and this is, applied for all generics. And they don't discriminate in the FDA approval between the narrow therapeutic index drugs, like the immunosuppressive medication, and we will come to this in a minute, and the general medications. And you can see that if a patient who has got, say, for example, a generic with the approved already, with the 80% of the Cmax or AUC compared to the branded one, and then because of the cost implications, this month they have changed it to another generic, which is already approved with the 125% compared to the branded one, then this patient will be exposed to around 45% excess in the active ingredient, with the potential of the nephrotoxicity if we are talking about the CNI. And you can inverse this equation and say the vice versa. He will be exposed to 1/3 less, with the problem of the risk of rejection. So this wide range of the, of the comparability to the branded makes some people very anxious, particularly the transplant physicians, that we might be exposing the patients to untoward hazard. That's why the EMA, which are the European medical agencies, and the Canadian authorities they have tightened this confidence interval, and they have put the range between 90 and 111%. Obviously, these used the Cmax, the concentration maximum, and the area under the curve to reflect the rate, the extent of the absorption, and the extent of the exposure of the patient to the drug across the time, which could be comparable to, bioavailability-wise from the pharmacokinetic and to the pharmacodynamic, and you don't need to do any clinical trials to prove the efficacy or safety. I have already explained this example about the wide range and the implications and the risk of this wide range with the nephrotoxicity or the potential of the rejection. And again, you can see here, you are looking at about 80% to around 125%. What are these narrow therapeutic index drugs? These are the drugs when the range between the subtherapeutic level up to the toxic level is very narrow, and you might need to have a drug monitoring. The classic example for the immunosuppression will be the cyclosporin, will be the tacrolimus, everolimus, or sirolimus. The mycophenolate, they are not a narrow therapeutic index drug, and you don't need all these tightened criteria, and the same for the azathioprine. What are the other challenges claimed for the generics? So the sample size to approve them, very limited, 24 to 40 patients. You are applying them to healthy volunteers, not to the transplant population, with the legacy of the transplant populations, for the absorption and for the pharmacokinetic and pharmacodynamic differences compared to the healthy volunteers. They are, single dose, which means that it doesn't give you a steady state of the drug, not over weeks or months. And you don't need a long interval to see or test the safety or the efficacy of the drug. It's just only a very short interval to prove the, the bioequivalence of the drug. Again, you don't have a comparison between the generic and the generic, and it is a common practice to switch from one generic to the other without consulting the patient even. I'm not sure whether this is the same case for the immunosuppression in the United States or not. The complications of the rejection and drug toxicity could outstrip, obviously, the cost savings if it is not rigorously controlled. This has, reduced the confidence of the generics between the public to the point that this is a very famous book, has been published by Catherine Eben, and I would encourage you to look into the YouTube about this, is around 40 minutes. It is fortunately about the generics as a whole, nothing to do with the immunosuppressive medication, and she has raised her concerns and her experience with the FDA approval for most of the generics who are coming from India and China, and how, frequent to find fraudulence from these companies or from these, manufacturers. And she, advises any patient when he comes to have a generic to check what is the manufacturer, if there is any warning sign from the FDA or not. I would, as I'm saying, I have had a listen to this YouTube, very interesting. What are the other differences? As I've mentioned, the generic might not be a concern in certain disorders like the cardiovascular, but when we come to the immunosuppression, the critical use, particularly if we are talking about a single functioning organ, such as the heart or the lung or even the liver, if they lose it, they lose their life. But if we are talking about something like the kidney, still we have got the dialysis. But for those patients, if they lose their graft because of the generic, then they will lose their life. We have spoken about the FDA approval process. We have spoken about the Canadian one and the AMA, and the S-type in NICE, which is the National Institute of Clinical Excellence in England. They stated clearly that the bioequivalence doesn't mean all the time clinical equivalence in practice. So if there is any evidence to use, because now you can see that the generics being used on a very huge scale across the world, not in particular region. So if there is any evidence behind, our current practice, Unfortunately, good quality evidence for generics is lacking simply because you don't need to have a well-designed, well-powered randomized controlled trial to have the approval. You just need the bioequivalence studies and the data from the bioequivalence studies. The safety and efficacy evidences are not required, as we mentioned, and that's why they are very cheap. The bioequivalence in healthy volunteers, not transplanted patient, is required for approval. Again, it downgrades the efficacy and the safety of these medications for the transplant population. This led the transplant professional societies to put their opinion. But if you can look into that, I mean, the generics, say, for example, for CNI, started from the 2008 for the tacrolimus, for the cyclosporin was around 1995, and the transplant professional societies was in 2011, 2012, and there is nothing that much has changed since then. And they didn't deny completely the use of the generics, simply because they are used widely, but they have put some recommendations to have to use these generics in a very wise state. So the European Society for Organ Transplantation, the ESOT, said that repetitive substitution of the generics is prohibited in the transplantation, and if it is being done, substitution should be made under the strict supervision of the treating physician or clinical pharmacist. The Canadian one in 2012, there is no insufficient evidence about safety and or efficacy, and is not recommended in pediatric transplantation, and close monitoring is recommended. They haven't updated this recommendation for the last 10 years. And this, I mean, concur with what I have started with in this talk, that I don't know whether there is any more to add or not. It's just only for the simple discussion whether we need to use it or not, and if we need to use it, what are the, the precautions that we need to adhere to when we use it. So neither of these societies prohibited or denied, but set precautions and standards 9 years ago. We will go to, two systematic reviews about the use of the generics in the field of transplantation. One by Molnar in 2015 and one by Kahn in 2020. The Molnar one, the drawback about Molnar and the Kahn one, Molnar has based the outcome on acute rejection, but the diagnosis and definition of acute rejection was based in a combination of both the biopsy proven or clinical grounds. So, you can't rely that much on something like this because you don't have the biopsy proven rejections. But Kahn, a systematic review and meta-analysis, was based purely on biopsy proven rejections in their outcome. The number of studies in Molnar systematic review were around 50, around 17 of them randomized controlled trial, 18 of them observational studies, and 15 of them were around non-randomized but interventional studies. Kahn et al. With around 17 studies, 11 of them kidney transplant, 5 for the liver, and 1 combined for the kidney and liver. What is interesting also as a difference between Kahn and the Molnar, Kahn has used the conversion and the switch of the generics, and also used the de novo use of the generics instead of the branded ones. Molnar didn't differentiate between both. Okay, the generics used in Kahn were 5 for the tacrolimus generics. In Molnar, they are around 4 for the tacrolimus. What about the outcome in the systematic review of Molnar for the neural versus the generic cyclosporin? No significant difference in serum creatinine after 6 months and longer between randomized controlled trials. And the odds ratio, the P to odds ratio was non-significant. If you can look into it, it doesn't favor the branded one neural versus the generic ones. Okay, and as you can see, it was 1.23 the odds ratio for the randomized controlled trial, and for the observational one was 0.66. 1.23 means that the neural probably has got a slightly higher risk of having acute rejection, but for the observational one, probably the, sorry, I've just mixed up. The first one for randomized control trial, the generic has got slightly higher risk of rejection, but in the observational one, the neural has definitely better and good outcome with the odds ratio of 0.66, which means that the chances of having acute rejection when you use the neural is around 60% compared to the branded one, or 66%. How many graft, loss is around 17 graft loss, but not all studies have documented, the graft loss. Some of them they have, some of them they didn't. But in Khan, most of them they have reported about patient and graft survival. There was no significant difference in serum creatinine between both groups when you use the Prograf against the generic tacrolimus, and the p-value was 0.7. For the cell set, so we have used the neural, there was no significant difference, whether it is a randomized control or observational. When we used it for the Prograf, there was no significant difference in the incidence of acute rejection. When we used it on the cell set, it is the same. There was no significant difference. Come to the clinical outcome in Khan trial, which is 2020. It has been about the kidney and liver, de novo and conversion, included only biopsy-proven rejections, which is the hard outcome, which we can rely better than the previous one. And they have stratified occasionally by the organ transplant, whether it is a liver or a kidney, and stratified also by the whether it is a randomized control trial, a conversion or a de novo. So if we can just look into the clinical outcome for the biopsy-proven rejection, stratified by the organs. For the kidney, the risk when you use the generic versus the branded, the risk is 0.93, which is in favor of the generic. And again, for the liver, it was 0.65. Again, it is in favor of the generic compared to the branded one. If we look into it stratified by the organ, by the de novo or conversion, if you use it as a de novo, the generic risk is 0.75, which is definitely better than the branded. And if we are going to do the conversion, the risk is 1.93, but statistically was not significant. So the risk for developing acute rejection was higher if you would use the conversion. But look into something very important. How much is the number of the patients that we use to just, drag or get this, statistical analysis? Very limited number. The maximum is around 400 patients versus 200 patients. And this was not even a randomized control trial with the risk of bias, inherent bias a lot. And again, the total number, if you look into it, doesn't count for a well-designed randomized control trial. And I'm not entirely sure the significance of the statistical analysis of these studies. But this is what we have got. And as I'm saying, we will not have, or we will not see a well-designed randomized control trial to compare the generics versus the branded. And all we have to rely on is our personal experience or, institutional experience. So back again, as we can see from the graft survival, they are comparable, whether it is on the level of the kidney transplant or on the liver transplant. Both were about the de novo studies, nothing to do about the conversion ones. Nothing has been reported about any patient loss in these studies due to this conversion or due to the de novo use of the generics. One of the studies has reported about the EGFR, which is interesting, but for 6 months only, not a long-term follow-up. And you can see that when they compared one of the generics, which is the Adaport compared to the Prograf, they have been, both of them after 6 months of the transplant, they have been comparable. There was no significant difference between the branded or the generic. For the histological differences, they were on the whole comparable to each other. There was no statistical difference between any of them. But if you look into some of them, you can see the EFTA has been slightly raised for the Adaport, which is the generic, compared to the Prograf. And again, for the borderline, it changes higher for the adipose compared to the Prograf. But if you look into the subclinical rejections, when you don't have any diagnosis of the rejections, you can see that it is more for the branded one, the Prograf versus the adipose. The normal one, it has been more in the adipose compared to the branded Prograf. Looking into the BAMF scoring, if you look into the BAMF scoring at 6 months of the transplant, you can see no statistically significant difference between both groups, whether it is the adipose or it is the Prograf. So if you look into these two systematic reviews, we can drag this conclusion. The sample size was small, risk of bias was high. Many of them, they have been observational studies, or at best, if it is a randomized control trial or interventional one, it was not well designed. A parallel form of randomized control trial might be an intervention as a crossover study. The follow-up for the, for the patients were very short, up to 6 months, maximum 2 years. And obviously you can't get from this, any follow-up for the chronic allographic nephropathy. Trough levels, obviously, in some of the studies, which is not identical to the AUC or Cmax, so you can't test for the bioequivalence, but this has been tested already for the approval anyway. Few randomized control trial, as we mentioned, inconsistent reporting about the rejection, because in the Molnar one, you've been relying on the clinical diagnosis of rejection in some studies, but in Khan systematic review, it was, purely biopsy-proven rejection. Some of the generics we've been in this systematic review is no longer used, has been withdrawn from the market, like the cyclosporin A, cyclosporin A, the Apple 4, has been withdrawn for some manufacturing problems and lost confidence in this particular type. And that's why they have taken it, the FDA has taken this from the market. Are they equal, the generic versus the branded? We can't put it in this way to say that they are equal. There is no good quality evidence to support either way. We can't say they are both equal. We can't say equally they are not equal. What are the recommendations of the transplanting societies, whether it's KDIGO or the European International Society of Heart and Lung Transplantation or the Canadian one? The conversion should be avoided or limited to specialized transplant physicians. Only the specialized transplant physician, they can do that. They don't deny to do it, but under the strict supervision. And if there is any conversion, should be accompanied by strict follow-up and monitoring. The BTS in the British Transplant Society guidelines, generic immunosuppression shouldn't be used unless they have been shown to be bioequivalent and approved by the EMA, the European medical agencies. The kidney transplant should be made aware, recipients should be aware of the existence of the generics and the importance of not switching between preparations. Of note, in the UK, the patient doesn't pay any penny for the medicines. It is mainly the government. So you might not need to apply this to everybody. Drugs should be prescribed by brand name, even if it's a generic. You need to write down who is the originator of this generic, because there is a lot of the kidney transplant recipients should be closely monitoring after switching between generic preparations until a new steady state is established. What is the situation in the United States? We can look into this study in Colorado and even national survey about the change of the use of the generic. You can see that the generic substitution for TAC and MMF for solid organ transplantation increased in the first year between 2008-9 up to 2010 by around 50%, and from this 50% from 2010 up to 2013 rose by around up to 78 to 81%. So you can see that it is completely has replaced the branded one. If you are talking about 81% of the tacrolimus and the MMF around 90% up to 2013, This means that most of the transplant recipients, they are prescribed the generic ones rather than which generics that you need to use. You can look into this curve very clearly from 2010 up to 2014, the huge increase in the use of the generics compared to a huge reduction in the use of the branded. And obviously 2009, here when the patent of the tacrolimus has expired. It did this transfer of the my kidney transplant patient in 120. Coming to the local ones for the British Transplant Society guidelines, you can see that generic immunosuppression shouldn't be used unless they have been shown to be bioequivalent and approved by the EMA, not FDA. You have to take into account that in the UK, the patient doesn't pay any penny for the prescription of the transplant. As we are saying, recommendations by the BTS, the British Society, is the generic immunosuppression shouldn't be used unless they have been shown to be bioequivalent. Use the criteria of the EMA, not the FDA, for their use. And be careful that the British Transplant Society, they recommend the generic immunosuppression where the patient doesn't pay any penny for the prescription and the government will pay everything. That's why they are very rigorous when they are using the generic. They also recommend that the kidney transplant recipients should be made aware of the existence of these generics and the importance of not switching between the preparations without appropriate supervision, simply because they can change from generic to the generic by the treating physician, and we don't have that much experience about the different generics in a clinical, evidence. There is no much of the clinical evidence between the different generics. Drugs should be prescribed by brand name, even if it is a generic, just to make sure that the manufacturer is well known and there is no warning sign from any of the regulatory authorities about this company or this manufacturer. The kidney transplant recipient should be closely monitored after switching between generic preparations until a steady state is established. United States nowadays, there is an interesting study about the consumption of the generic medications for the kidney and liver and heart transplantation nationally and in Colorado. And what they found for the TAC and the MMF in particular has increased. The use of the generic substitution has increased substantially following they introduced it in 2009 for the tacrolimus. It increased, as I mentioned before, in the first year from 2009 until 2010, increased by around 50%. And the following 3 years, from 2010 until 2013, it has increased to be around 78 to 81% from the total tacrolimus available, all generic. For the MMF, it reached around 90% by 2013 or beginning of 2014. You can see here the curve. It's very clearly for the kidney when the patent of the tacrolimus has expired in 2009 and they start to use the generics. The generic use has increased exponentially until it reaches around, as I'm saying, around maybe 75% by the beginning of 2014. My personal experience, I have changed around 120 patients or converted 120 patients from the brand Prograf into the generic Adaport, and I didn't lose any graft for these 120 patients after 1 year of the switch. When I have just made the change, it was like for the dose, and I have done the trough level check a week later. I didn't find much of the swings of the trough level. It was, comparable, although I didn't do a statistical analysis, and the dose adjustment were within the, the dose adjustments were within the same confines that are usually used in the routine practice. I have saved around 48,000 sterling pound, but I didn't include here the drug monitoring, which was an extra single drug monitoring after the switch. The conversion included kidney and the combined kidney and the pancreas. I can't say anything about the liver transplant. I receive many children with a kidney transplant. They are already on the other boat. For the patient who is paying from his own pocket, probably like in Egypt, there is a greater chance for better adherence when they can tolerate, the payment of the prescription. And this obviously will be reflected in a better graft survival. Thank you for your attention, and I'm happy to receive any question to answer it. Thank you.